Welcome to the Thor Hour. I'm your host, John Thorhauer. Join me in conversations as I learn from experts, enthusiasts, artisans, and other folks as we consider the human experience. Grab an episode or two and explore the talents, passions, and curiosities from Frederick, Maryland, and beyond. If you enjoy these conversations and want to help me out, please like, subscribe, and share with your friends. Today I'm speaking to Miranda Darby. Miranda is the assistant professor of bioinformatics at Hood College. Miranda has a Ph.D. in molecular biology and genetics from the Johns Hopkins School of Medicine. She has a passion for studying the human genome and teaching others to do the same. Join us in this conversation as Miranda speaks about the challenges and possibilities bioinformatics holds for us. I hope you enjoyed this conversation with Miranda Darby. Well, Miranda, welcome to the show.
Thank you.
Yeah, thank you for coming. Really happy that you could be here.
I'm happy to be here.
Yeah, and the topic, bioinformatics, is I'm really interested in the topic. Super exciting, but it's also one of these topics I've found a few friends of mine, as I've mentioned the show, we're going to be doing this recording. I say bioinformatics and they start scanning like, wait, what does that mean exactly? Yeah. And so maybe like the first thing you could do is just like share with us, what is like basic kind of like idea of what bioinformatics is, and then, why is it important? Why should we care about it?
So there are two very closely linked fields, bioinformatics and computational biology. Okay. And they're really just two different ways of looking at the same thing. Both of them involve using custom computer or advanced computational tools to analyze biological data. And quite often, especially since about 2010 or so, the big boom has been with big data, biology, right. Big data. So that's mostly next generation sequencing data that we're talking about with biology, right. But it can be mass spectrometry or other types of data as well. But mostly it's next generation sequencing. So it's sequencing RNA or DNA. And it used to be, years and, hundreds of thousands of dollars to sequence one person's DNA. Yeah. Now you can do it for a few $100 in a day or two. Yeah. So
it's just opened up the possibilities.
Exactly.
For what can be done, but also who can do it.
Exactly.
Right. Huh. Okay.
And just to clarify, bioinformatics is really the field of developing these computational tools and algorithms and, getting them up and running. Okay. And computational biology is more being an advanced user of these tools. So not necessarily developing the algorithms, but understanding exactly what the algorithms do and how to interpret them biologically. So if you see certain numbers, what does that actually mean? How do you interpret it?
Okay. So when did bioinformatics, it's not, when I went to college, I don't remember hearing about bioinformatics. Maybe something was there like that, but when did it become a field of study?
I'm not sure.
Roughly,
I mean, the very origins of it were actually in like the 80s.
Okay. I mean, that's not long. When you
no,
think about
it's not.
like
Yeah.
You go away to study in college and you get degrees in. That's not very long.
But the way we think of it now really didn't become a field until like 2010 or later.
Oh, wow.
When I was in graduate school, even, of course, I'm a little old, but I went to Johns Hopkins and I did biochemistry, cellular, and molecular biology as my field of study. And we had a bioinformatics course where we didn't use any computers. It was all just about these algorithms that they were just had kind of recently been developed to find proteins and align protein sequences to each other. The original matrices were developed in like the 60s and 70s, but then trying to...
So you say matrices, that would be like the science or the math behind it?
Yeah, basically the math for how to align one long string of protein, of letters to another long string of letters. That was developed, I think, in the 70s and 80s. Maybe even starting in the 60s, I'm not sure.
Okay.
But it's now been expanded and expanded and expanded. And by the 80s, what they had done is find ways to identify patterns in proteins. And as sort of a string of letters, and they had started making databases of these patterns, of these different signatures that you could find in proteins.
So for people who aren't scientists, is that could we think of that sort of like if I have my photos in Google Photos, I can search for dog, and it can find all the dogs. And it didn't used to be able to do that, but now it can bring up all pictures of dogs. Right. Kind of something like that, pattern matching,
much like
kind
that.
Of a thing.
Yeah,
which is super powerful, right?
It is, and that's what really started going. And even when I was in graduate school, I graduated in 2012. But at the beginning of graduate school, like I said, we did a blossom matrix, which is a way to figure out how good a match two proteins are to each other.
Okay.
But we had to do it with pen and paper.
Wow.
But
it'd take a while, right?
Oh, yeah. And really, there's no reason anyone needs to fully understand that, to be honest, in order to actually run it. But that, for instance, any biologists who look at this will be familiar with NCBI BLAST. So the Blossom matrix is one of the algorithms that BLAST uses to find.
So with bioinformatics, can you give us a sense of how broad? It seems like the field is very broad.
Yes.
Right. Like, do I understand correctly? Like, do both of us have watches that kind of do a version of bioinformatics? Like, it's taking information that's biological and reading it. Is that bioinformatics or not?
It could be. It's really. There's all of these terms that get kind of included. So one thing that we're all moving towards is precision medicine.
Yeah, that's amazing.
Which involves a lot of bioinformatics, but it also involves some health informatics. So the way we tend to break things down is bioinformatics is specifically looking at biological data. And health informatics is quite often looking at clinical data, or even biometrics, like our watches are gathering. Right. But certainly you can use biometrics in bioinformatics as well. It's all quite fluid.
Biometrics.
So biometrics is just term. Any sort of measurement of a biological phenomenon.
All right. So within bioinformatics, you said it's within the study of things like in the human body, right? So one of the things we'll probably talk a lot about, I assume, would be genomics. But then there's also these studies of cells and multiple cells, right?
Yes.
What is that called? And
so
how is that? That's
this has
really
all
different, right?
Been this evolution. So like I said, originally we started out with just some databases of key sequences of proteins and adding to that. And then since the advent of next generation sequencing, now we have terabytes and terabytes and terabytes of sequencing data that of both humans and organisms that's de-identified and freely available for people to look at. And that's just been snowballing. I mean, I'm not sure exactly when those things started, but the amount of data in the major repositories for sequencing data has been outpacing Moore's law.
Oh, really?
So far as so actually a big problem has been having enough storage for all of this data.
I could see that.
So my last major sequencing project was 6 terabytes of raw data.
Wow.
And that was the raw data.
Yeah.
And then, all your analyses
Right.
take up space
Just
and that sort of
build
thing.
on top of that.
And so to do that, you start to have to learn all of these skills. So I was a molecular biologist. I had no programming when I started.
Right.
I taught myself Linux, and
You first started, you're like megabyte, gigabyte, terabyte. What's the...
yeah. I mean, I learned a tiny bit of Perl as to just kind of get started, and then that quickly became no longer useful. But I learned Python and R and Unix, Linux, and. So, some would argue that that's bioinformatics. Then when you're able to use these remote compute clusters and do things, you can't do a major next generation sequencing analysis on your own laptop.
Right.
Because it just doesn't have enough power.
Yeah, or space or anything. Right?
But you can use online tools, or what we typically do is use a remote compute cluster. So it's basically a big computer with a bunch of hard drives and a bunch of processing cores.
Right.
And you kind of just interact with it through your own laptop.
So you mentioned something that's real foundational. So maybe you can help explain it so we all understand, because we hear it a lot. And I know it's important, but what is it exactly, which is like sequencing? So what is sequencing? And why is that so important for us? Because you've already used the word a whole bunch.
Yes.
Right? Like it's just a foundation of what you do.
It is the foundation of what I do.
Yeah.
And it is very much where my interest in bioinformatics has fomented.
Okay.
But sequencing, I guess at its most basic level, is finding the series of, in DNA, A's, T's, and G's. Okay. In RNA, it would be A's, U's, and G's. And for proteins, it's the sequence or the letters for the amino acids. So for proteins, they're long chains of amino acids, and so you can name one. Each amino acid has a name. Okay. And so if you see a certain number in a sequence, there's actually databases out there where you can identify a large number of proteins from just a small number of proteins in amino acids in a sequence. Then you can look that up, and if it's unique to a protein, you can find that out.
So sequencing is literally that base. It's that simple.
It's that simple. It's finding the sequence
each of the letters that we say A, C, T, G. What order they're in, and maybe how they connect up, and then it grows from there, I guess?
Well, yeah, so DNA is, do you have any idea how many A's, T's, and G's there are in DNA?
No, I have no idea.
So there's roughly 3 billion.
Oh, okay, wow.
And do you have any sense of what percentage have a known function that we know what they do?
I think it's small, right? But I don't know the number.
It's between 2 and 3%.
Okay, it's even smaller than I would have guessed.
Yeah.
Okay.
At least between 2 and 3% are known protein coding genes. So what we think of as a gene is a section of DNA that codes for a segment of RNA that gets spliced and chopped up and put together and then read by these things called ribosomes. Okay. And that makes the sequence of the protein. Okay. So there's a linear relationship between the DNA and the RNA and the protein. Okay. If you know the DNA sequence, you can kind of guess what the RNA, you can certainly guess
how it's going to be used.
Yeah, you can guess what the RNA is going to be if you know where the RNA is going to start and where the RNA is going to stop. Okay. But you also have to know where all the little chunks that get cut out are. Okay. So a DNA for a gene might be, millions of bases long, and the final RNA might be thousands of bases long, because there can be things cut out. Now, usually the difference isn't that big. Yeah. Usually it's like a tenfold difference or so. But
so I think you were getting close to the term, that we hear quite a bit, which is junk DNA.
Junk
right?
DNA,
is that
yes.
at one point, the other, whatever, 97, 98%.
Exactly. And even when I was in graduate school, we were taught it was junk DNA.
Yeah.
I never actually believed it.
That term seems really strange to me. As I thought about it, I looked it up to try to understand a little bit like where it came from. And I don't think they know exactly, but I read a couple papers where people tried to kind of figure out, where it came from. And some people seem to trace it back to maybe even the late 50s. But wherever it came from, it just seems strange because these are scientists, right, calling it junk DNA. And like, you'd think they would assume maybe they just don't know,
right,
rather than it's probably junk.
Well, I think we tend to have contempt for things we don't understand, or at least a certain proportion. Some of us are in awe of things that we don't understand. I love junk DNA. That's really my, what gets. Or something. I should. I really should. My students, of course, any of my students who see this are just going to be laughing because I spend a lot of my lecture time talking about how much there is left to discover. Right. And in fact...
Yeah, because if you assume, which we have, why not assume it, that the other 97-90% is meaningful, then there's a lot of meaning to find.
Right.
Which is incredible to think about, right? I mean, that's like if somebody said, we've discovered 2% of the land that there is to explore on the earth. People would be going out to find that other 98% and say, what's it like out there? Right. And so that's what you're doing.
That's what I'm trying to do, yeah. I mean, not even I think that everything is meaningful right now. But what we have is a legacy of millions of years of evolution. And I think there's an evolutionary benefit to keeping some of that DNA that we're not using. Okay. So remember, we have 3 billion base pairs of DNA. Yep. Roughly 50% of that is actually repetitive sequences and other junk that are left over from endogenous retroviruses. So you know how HIV can hop around and be in your cells? Well, that's an exogenous retrovirus. There are endogenous retroviruses as well, most of which aren't actively hopping around in humans anymore. But over millions of years, they've left kind of patterns. So these are chunks of DNA that can replicate themselves or make an RNA copy of themselves and then a DNA copy and then insert themselves someplace else in the genome. So now there's 2 chunks that match in the genome. And so roughly 50% of our genome is made up of that kind of stuff. Wow. And the current theory is that these are, some of these can be used as a way to rapidly evolve. So if you think about a repetitive sequence that's spread throughout the genome, if you think about a protein such as a transcription factor, transcription factors are proteins that bind to DNA. And when they bind to DNA, they attract the machinery that causes RNA to be made from it. So they're very important. They basically control what RNAs are being made in the cell at any given time. But they have certain binding sites that they identify. And if you imagine that one was to mutate so that it identified, instead of its regular place, it identified a repetitive element place, then all of a sudden there'd be hundreds or even thousands of new places that would start to be transcribed.
Like instantly. Because they're there.
Right.
They're waiting, so to speak.
And a lot of them then might be detrimental or might not be good, but some of them might. Right. And over time, what we've found is that, in fact, that does happen, and there are transcription factors that have taken over and recognized the legacy of some of these repetitive elements and made whole new regulatory structures, because a transcription factor, if it's binding to 100 different places, those 100 genes are going to be co-regulated. They're going to be up and down at the same times.
So is there a way, is there a branch that is trying to, like experimentally predict like what would have happened? Like go back and say, we assume this would come out of this kind of a repeating sequence with that scenario and like testing it?
People are doing that, I believe.
Yeah. Okay.
Another thing that people are doing, and I can tell you a little bit about what I've been doing. What we were trying to do, actually, I was working at the Stanley division at Johns Hopkins, and we were studying schizophrenia and bipolar disorder. And there are no genes that, well, there are a few genes that are reliably associated with schizophrenia and bipolar disorder, but they don't give rise to most of the disorders. So if you have a mutation in the relin gene, chances are very, very good you're going to have schizophrenia.
Okay.
But if very few people who have schizophrenia have a mutation in the relin gene. So there's a lot of different potential causes of schizophrenia and bipolar disorder. And anyway, so people had been looking for genes for quite a while, and we decided they'd also been looking in the DNA, and they found a lot of just marks in the DNA. Basically, they're just little single nucleotide polymorphisms that mostly don't do anything. They're just little differences between your nucleotide sequences and my nucleotide sequences and so forth. And those little differences you can map to certain traits. And if you have hundreds of thousands of people, which is what they've done with schizophrenia and bipolar disorder, you can sequence, many, many people's either their whole DNA or just the exome, which is just the 2% we know does stuff. But whatever, they've done a lot of DNA sequencing, and they've found that there are 108 different spots in the genome that correlate with schizophrenia. But nobody knows why, or what in that region. And some of those regions are quite small, maybe 1 or 2 genes. Some of those regions are like have 10 or 20 or more genes. They're huge, so it's hard to tell. But
there's a correlation.
Yeah, there's these correlations. But when you really look, fine-tuned, none of these correlation places are actually in genes, or very few of them are. Most of them are between genes or in introns. So remember I said an RNA gets made and then it gets chopped up and put back together?
Yep.
The introns are the part that gets chopped out and discarded.
Okay.
And the exons get put back together.
Okay.
And anyway, because there were some DNA studies that showed that there's some missing heritability in schizophrenia and bipolar disorder, we decided to look at the RNA. So DNA is going to be the same in every cell. RNA is only going to be made where it's being used, theoretically. And we decided to see not just looking at protein coding genes, but any RNAs that get made. Where are they coming from in the genome? Are there any that are coming from these areas that are between genes or, in introns? And we found, in fact, we looked just at repetitive elements because they were strongly thought to be junk DNA at the time. And, we just thought, well, let's see if there's anything here. My colleague, Sarvan Sabansian, had already done work to show that certain of these repetitive sequences had increased expression in schizophrenia and bipolar disorder, but we didn't know, if there's 100 copies of a sequence, we didn't know which one of those 100 copies or all 100 copies, if they were all being turned on. So we worked with some people at Johns Hopkins to find ways to uniquely map RNA sequences, even repetitive RNA sequences, so we can identify where in the genome they came from.
Wow.
And without trying to get into too much detail, what we found after a lot of surprise and stuff is that many of these are being expressed, and then when we went to look at the context for how they're being expressed, and we were looking at human brain RNA, we found that they're actually novel exons of protein coding genes. So they're genes that we know about, but quite often there would be a repetitive element that was in an intron, which is in a part that usually gets cut out and discarded.
Right.
But it was actually being spliced in and continued and included in these protein coding genes. So that's been a new form of study. And what I've been doing with my students at Hood has been to work on identifying tools to determine if you see something like that, how likely is it that it's going to be functionally important? How likely, if you only make a few copies of something, that can just be junk that, it's not worth the cell's wherewithal to stop doing that.
Okay.
But if something's being transcribed a lot, then that's taking energy from the cell. So that's more likely to be functionally important and so forth. So anyway, that's part of how we're finding some new stuff in the junk.
Okay. So you mentioned, Hood there, which is where you are now. What is the focus at Hood with bioinformatics? Like, what are you all doing? And I'm doing the research on identifying novel transcripts and trying to identify whether or not they're functionally significant. Okay.
And I've been working with a number of different students on that.
Okay.
And then there's another one of my colleagues who actually does what's closer to health informatics. Actually, a couple more of my colleagues are working on something that's closer to health informatics, looking at working with data from the military, actually, and tracking. I'm not sure. Actually, it's all kind of quiet, but I believe part of what they were looking at was PTSD insults and how that correlates with all kinds of different exposures and things like that. Okay. But don't quote me on that. I'm not sure it's PTSD. But they've been looking at various things. And then we collaborate a lot with people at the NIH.
Okay. That's great.
So many of our students do capstones or theses at the National Institute of Health.
That's pretty cool.
Yeah, it's very cool.
Yeah.
And many of those are at the National Cancer Institute. So they're looking at novel animal models of certain kinds of cancer, or there's all kinds of things you can do.
Yeah. So one of the things when we were talking earlier and just trying to get a sense of what you all are doing, and me trying to understand a little bit about bioinformatics, I think you had mentioned before that some of what you do at Hood is akin to like creating new tools. Mm-hmm. So talk about that a little bit. What is that, and why is that important?
So the vast majority of molecular biologists need the information you get from bioinformatics in order to do their jobs these days. Okay. Which is actually part of why we about 20% of our students already have PhDs, mostly in molecular biology and genetics, which is what my PhD is in as well. And they're coming and retooling and learning new tools because they may not need to actually do next generation sequencing, but they need to understand it and need to be able to interpret it for the most part. Okay. And even if you're farming something out, you need to be able to know whether you can trust it or not. Yeah. And so I'm sorry, I got a little off topic here because my mind's just firing. But one of the major issues with bioinformatics right now, and has been for the last 5-6 years, well, actually since its inception, is there are no gold standards for how to do a lot of things. So, for instance, if you're trying to, so when you do sequencing, most people do short read sequencing, which means you have these reads that are about 100 bases long. So, 100 ACTs Gs long.
Yep.
But you have 240 million of them.
Okay.
And so using computational tools, you put them together and figure out, how they all fit together into something long.
Yep.
But for RNAs, you don't necessarily, so for DNA, it starts at one end of the chromosome and it goes to the other end of the chromosome.
Mm-hmm.
And if you have a chromosome to match it to, it's pretty trivial to just align things to the reference chromosome and figure out. But if you're trying to look at RNAs, those start someplace and end someplace and have gaps in the middle.
Mm-hmm.
And so the tools to try and figure out those are really hard, because you might have, this is hard to do without visual aids, but you might have an RNA that has exons 1, 2, 3, 4, and 5.
Right.
And another exon that just has, another protein from the same gene that's just exons 1, 3, and 7.
Okay.
And another one that's just 1, 2, and 4. And they're all being made from the same piece of DNA, but they make different proteins. And trying to figure out which of these collections of RNAs are being made from the same DNA is extremely difficult unless you happen to have that golden 100 base pair piece that crosses from one part of one exon to another part to another exon. Then okay, these two were together and like that.
So when you talk about a tool that can help you understand that, is the tool like code, it's an algorithm, or is it a piece of data, or is it both of those things?
It's both of those things.
Okay, so it might be an algorithm that has worked with a set of data that when you apply it to your stuff, it'll illuminate that and you can understand it better.
Right, exactly.
Okay.
Yeah.
Interesting. And so these tools, are you have like this tool chest, but your tool chest. And then if I were to go down the street to somebody else is studying, their tool chest might not necessarily be the same tools, right?
You have the problem. Yeah, that's exactly right.
But you might both be trying to study similar things.
We probably are trying to do the same.
But different tools.
There's a lot of reinventing the wheel.
Yeah.
And there's a lot of nobody knows whose tool is better.
Yeah, right.
Because many of these tools are trying to do things that are so computationally difficult that we only use what's called heuristics, which means we have sort of an approximate answer.
Yeah.
Because a lot of problems from a computational standpoint are NP complete. I don't know if you're...
I have no idea.
It basically means it would take you forever to try and... Like if what you wanted to do was use trial and error and brute force it and try every possible way to do it, it would take you a million years. Okay. So you can't do that. Right. You have to make things that say, okay, I'm going to make certain assumptions. Yep. And statistically. Statistically, I think it's more likely if I do this. Okay. And everybody has different ideas as to how that should work.
So do you think there'll come a time where kind of the cream rises to the top?
It's already happening.
Yeah.
It's already happening for sure. And
what does that look like when that happens? Like, does one group of people who's studying, all of a sudden the tool that they're using just produces this answer and it's more clear than anybody's ever seen before? Or like how does that come about sometimes?
Sometimes it's more accurate. Sometimes it's faster.
Okay. But do you know, like, when it happens, like, oh yeah, this is definitely like we got the right tool, the right data set? Or...
I think if I were looking at it, I could. I'm at this point now where I'm running this program, so I'm trying to keep track of all the stuff that's coming out, and also, get my students internships and do all the other stuff. Yeah. But what happens is, over time, people just agree that one tool is better than others.
Consensus.
It just sort of becomes the tool that most people use. And there's there's certain labs that are good labs that have good reputation.
Right.
And so if they put out a tool, I tend to be more likely to trust it.
Mm-hmm.
And if they tell me don't use this tool,
Right.
I trust them on that
Right.
as well. Of course, I'm very privileged having gotten, I learned bioinformatics from people who are creating some of the best tools out there.
Yeah.
Even though, I don't consider myself a computer scientist, I'm more someone who uses tools as opposed to creates these tools.
But that speaks all to, like, what you just mentioned is something that struck me when we talked earlier, and I thought it was important for you to talk about a little bit, and that is how open, in general, this field seems to be.
Yes.
Which I didn't know I mean, I wouldn't have suspected that was the case.
Mm-hmm.
But for me, being in software development, there's a lot now that is open source software, but that wasn't always the case.
Right.
And it sounds like this might be akin to that. But it's not just like that, although this sounds very important, that the tools tend to be like, if they find a good tool, you share it. And you say, hey, this is a really good one. Everybody should be using this. But then also data sets, making that data available. So you want to talk about those things a little bit?
Sure. Yeah, I'm happy to. So from the tool standpoint, as you said, everyone's using a different tool, and it can be very difficult. And it's led to sort of a reproducibility crisis is where we got to several years ago, because everyone was using different tools and you get different answers even from the same data using different tools. And so there has been more of an effort to share tools and to make sure everyone's on the same page. And when you publish, to share not only all of your raw data, but also the code that you use to analyze that raw data.
Right.
That's being adhered to more or less depending on what journals you're publishing in.
Right.
And
so some of the journals
so
will
forth.
have a better reputation for that.
Oh,
perhaps?
definitely.
Okay.
But also, sometimes if you're publishing in a proprietary journal, then it might be more difficult to share everything you want to share broadly.
Yeah.
Although largely, the researchers that I follow tend to share stuff on GitHub. I don't know if you're familiar with GitHub.
Yeah.
So a big way.
So just to explain to some people who aren't programmers, or, I mean, it's a place where you can share code or really any information, but it's open to everyone. If you make it that way, you can make it open so that anybody can go in and they can kind of click around and explore stuff and see stuff and interact with you on it, right?
And lately, there with these tools, some of the great players in the field, from the very beginning, pushed open source and really kind of proselytized,
So
open source.
They kind of set
They
that
set
foundation.
this tone. And then also in the last 8 or 9 years, they've also been pushing reproducibility. So like one of my heroes in the field is Jeff Leake, who was at Hopkins. He's left now. But he has pushed a lot about reproducibility. And in fact, that's become one of the foundations of the program that I developed at Hood, is to make sure what we do is reproducible.
Which, I mean, having that kind of coming along, that shows the maturity of the field, right? Like if you're getting to the point where that's becoming important, that means you're getting to a very important level of maturity in the study, right?
Absolutely.
So,
So
that's a good thing.
It is a good thing
in a lot of ways.
And so there's this push to make things fair. And fair is, an
that's
acronym.
An interesting word.
We love our acronyms, but in this case, it's findable, accessible, interoperable, and I think reproducible.
Okay.
I think there's one other R too.
Okay.
But anyway.
Okay.
I'm not used to teaching without slides.
Yeah.
I'm used to having a little...
oh, you're doing great. No, that makes sense.
I do a lot of interpretive dance, actually, a lot of writing on the board.
Yeah.
I tend to have like all the walls of my classroom,
oh, really?
Written on.
Yeah. Keep things moving.
I burn a lot of calories while I teach,
right.
actually.
Yeah.
my watch always wants to log it as a workout.
Yeah.
But anyway, so a lot of really great core academic researchers push this idea of making things freely accessible and interoperable.
Yeah.
And it started out with some really big library repositories. So for those playing at home, a computational library is essentially a set of functions that you can use in various ways. So it's a set of functions and the rules for how to use them.
Yeah, it would be like handing everybody the same little toolbox.
Exactly.
Say, we're all going to get the same tools, that everybody, when you pick up a hammer, you're going to have the same hammer I have, and it's obvious how to use this hammer.
Exactly.
Right? Yeah.
That's exactly right.
Yeah.
And so they made a great one called Bioconductor.
Okay.
That a lot of top researchers around the world not only post their research to, their tools. I'm using this word. So a tool can mean, a database. It can mean a single function. It can mean a whole
broad.
big program. Yeah, I'm sorry I keep using that word,
that's okay.
but that's honestly what they are,
Right.
because some of these are very simple little things.
Yeah.
Some of these are really big, complex things.
And probably some of the complex things used to be smaller things that got put together into a complex thing.
Exactly.
Right? Yeah.
And basically the way bioinformatics works, because the data is always changing, because the instruments are always improving, and the way we use the data is always changing, because the other kinds of data we have available to compare it against is always changing. You have these little modules, these little tools, and then you kind of tape them all together, either using R or Python or Unix Linux. So, for instance, using Unix Linux, you can take a lot of command line tools. These are the kinds of tools you use at your C prompt.
Yep.
And you can run those and then have the output using Linux, have the output of one put into the input of another, and the output of that one put into the input of another one. So you can stitch things together using Linux pretty easily.
Oh, cool.
Or you can use R and take a bunch of libraries and put them all in your same workspace and use, this function from this library and this function from this library and put those together. And so those are the sorts of things that I do as a computational biologist.
Okay.
And then,
those are called, I think I've heard the term workflows used quite a bit.
Workflows, yes.
And that's becoming very important, because I could imagine, like everything you just described becomes perhaps a standard workflow to accomplish something, right? Like a standard way of chaining all these things together.
Exactly.
Yeah, okay.
And so then people start to publish their workflows, and then, things kind of gain market that way.
Right.
And by market, I don't mean that it's being purchased. I just mean it's being used.
Yes.
And that in itself is very important, because if you think about it, everybody was coming up with their own individual tool for every little thing, and then tying these tools together. That means only one group is testing each of these tools. Yeah. And usually these tools were just being tested on whatever data they had that they were trying to analyze.
Limited scenario, limited data set.
And if you consider that many of these programs were being created by students and postdocs who then graduated or moved on to the next place, then nobody's maintaining that tool and keeping it working. And so within a couple of years, that tool's broken, because the inputs aren't the same inputs anymore, and the outputs don't go into where they need to go anymore, and so forth. Yeah. And so in order to kind of keep the chaos from being so chaotic, that people have been pushing sharing all of their work through GitHub and other places, through Bioconductor, and there's also a Python community.
Okay.
So Bioconda is a Python community of people taking programming or bioinformatics tools essentially and making them easy to put together in a Conda environment, a Python environment.
Okay, so that's one of the things I wanted to ask you about, which you're talking about a little bit, with the challenges in your field. Like one of them sounds like you described a lot of things there, right? But it's like this quick, fast-moving environment without standardization, but moving towards standardization, and what does that look like? But there's these other challenges too, as I understand it, and you've already mentioned them a little bit, but it would be interesting to hear you talk about these some and how you approach this at Hood. There is a challenge, like you were saying, the average person, I can't on my laptop do this, right? So there's a computing power challenge. Like, how do you address that? And maybe, a little bit more on the sources of data and maybe how you at Hood address that as well.
Okay, happy to. So just as these tools kind of are developed by people, but now are being shared. Yep. And of course, as they get shared, more people are using the same tools, so then bugs get found and fixed, and the tools get improved. The same thing is happening with the data. Data is being shared to a number of central repositories. I guess I can't say a number of central because, that's... Right. Well, that's contradictory,
right.
But
there's some main ones.
But there's a few main repositories that, again, early on, people would consider microarray analysis to be bioinformatics. That's something we used to do on chips, these little printed chips. Okay. And instead, so some of my sequencing analyses, I've been working on data frames, or basically like your Excel spreadsheet, but with tens of millions of rows. A microarray is more like 20,000 rows. Okay. So it's still tiny, kind of big,
but relatively speaking, tiny
to my. I think it's small, and most people think it's big.
Yeah.
But that has largely how to analyze microarrays has been largely solved at this point. The tools have sort of converged on the same, to the same level where you get more or less the same output from the few major tools that are out there, and the considerations, like the different ways you have to adjust the analysis depending on exactly what microarray it is, et cetera, have also been worked out for the most part.
Okay.
That's sort of a solved problem. And, sorry, I'm kind of all over the place here. What got me on microarrays is when I first published microarray data, I actually had to include it, the entire 20,000 rows, as a printout, essentially. Like it was just a table, like a word table.
Wow. Okay.
At the back. People were so not thinking about bioinformatics and programming that in these biological journals, the way to submit that data was as a word table, essentially, with all of the data for all of the rows printed out. Yeah. And then, of course, I printed figures with, the data that I was highlighting. Yeah. But I included all of the raw data essentially as this giant word table.
That's crazy.
Which is, of course, not useful to anyone, right? Because even all those little symbols and things that denote which gene is which, there's 10 different versions of those symbols.
Oh my gosh.
There's several different versions of the genome even. So if you want to download, the set of all RNAs, for instance, there's 5 or 6 different places I could go to download the set of all RNAs, and none of them match.
Okay.
So basically, we're getting this way with tools and data the same way. We're getting the same way with information and data as we did with tools, where
Right.
eventually stuff started to converge.
Right.
I think, we're kind of converging on the same few
Yeah.
versions of the genome that everyone's using now.
But really, if you think about it, which I'm sure you have, but that is the way to get to the best stuff.
Exactly.
Like there is no other way.
Right.
Because if it were just prescribed ahead of time by one single group that for whatever reason either like had all the money or had all the power, you would never know if that was that the right choice or
Right.
not. But the way it's happening, the good stuff bubbles at the top because it's the good stuff.
Yes.
Right?
Yeah.
So whether it's the tool or the data or whatever, so it's been tested.
But it's also through a lot of good work from people all over the field. So
okay.
as you know, I mentioned a reproducibility crisis, and there was many years ago, not just because one tool doesn't behave the same as another, but there were a number of things that went on where people were kind of overstating what they could learn from the data.
Oh, okay.
Partly because they didn't understand the failings. And the field, many top academics in the field have really turned the field around by highlighting the need for us all to share, to do things reproducibly, to share our tools and play well together.
Right. About how long has that been an emphasis, would you say?
I mean, I'm sure other people were doing it before I was aware of it, but I became aware of it about 7 or 8 years ago.
Okay. Huh. Interesting. So...
sorry, just to
No,
finish
go
on the
ahead.
databases.
Yes.
So it used to be everyone had their own databases. Kind of like I said, I included all that data in the paper,
Right.
which is not very useful. And it used to be paper. Then you had to submit your sequences to journals, but then journals didn't want to be responsible for housing all of that data,
Right.
and so forth. And so what's become is there are a few private and a few public data consortia that really have done an excellent job of gathering all the data.
Yeah.
And so
Are
now
those international? Or
many
national?
of them.
Yeah. Okay.
But the way to access it is you have to have an NIH research number. So even this kind of it keeps people who aren't working at a university sort of out of it. Sometimes you can get the data, but often you can't get the data unless you have an NIH research number.
Right.
You don't actually have to win a grant or anything. You just have to be registered
Okay.
as a primary investigator, essentially. But you have to prove you're at
Right, you're
an
serious.
accredited
You're doing
college.
serious work.
You're doing serious work.
Right.
I'm not trying to do this for profit. I'm trying to
Yep.
do this to further science.
Yeah. Do you I don't know if this is the right way to even ask the question, but you just made me think about it with something like this. Do you have when you're allowed in, right? So you've been given that access. Is it read-only access, or can you write to this stuff?
It's read-only.
Okay, so
Yeah.
you can't mess the data up, I guess.
Yeah, no, you can't. You can only. It's usually FTP servers. You can just download stuff from.
Okay, all right. Well, so that's the data problem, which is interesting. I mean, so organizations, I guess, have chosen to take that upon themselves, and they're spending their resources to make that available. But then you still have the computing power challenge, right? So how do you all solve that?
So at Hood, what we've done is build our own compute resource.
Okay.
At the NIH, they have several different computing resources, but one of them is called BioWolf, and it's the one that the computational scientists at NCI tend to use. And that's where my students tend to go for internships and capstones.
Okay.
But it's a big computational server where you have to use Unix, Linux. You can't click. There's no windows. There's no icons.
Yep.
It's not the way people are used to doing things. And so we built our own at Hood so that my students could get used to playing in it. I call it the sandbox.
Yeah.
And you can throw sand in our sandbox and accidentally delete 2 terabytes of our data.
Right.
And it's embarrassing,
Right.
but I have it all backed up. But you don't want to accidentally delete 2 terabytes of data at your workplace.
Yes.
And
Yeah,
it's actually
imagine.
quite easy to do with Linux if you don't know what you're doing.
Yep.
A typo can cause that.
I've done that before. So, yeah.
And every bioinformatician I talked to when I was developing the program and asked, what do we need to have? They all talked about, but don't send them to me for an internship unless they know Unix, Linux, because they all have stories about,
because
whole projects.
If you're learning, there's a learning curve, and there's some pain in there somewhere.
Exactly. So we built our own. And it mimics the way BioWolf works.
Okay.
So that
was probably a fun project.
It was actually. It was challenging, but we have a great sysadmin.
Okay.
When I first started, we didn't. So it took a long time for me to kind of convince everyone this is what we needed and get it up and running.
So it's a big investment.
It is a big investment
of resources, expertise, money.
Exactly.
Yeah.
But we got it going.
That's really cool.
And we
exciting.
keep updating it with the latest software as stuff keeps coming out.
Is that a combination of people who are on staff at Hood who keep that thing running and students, or how does that work?
Students break it and staff and I fix it.
Okay. So you're still pretty involved with that?
I am. At first, I was the one doing it all.
Yeah, it sounds like it.
But now we've actually gotten it more complicated than I know how to do.
Yeah.
So at first, we had a very simple system because I was the one doing it. And like I said, I'm a molecular biologist.
Right.
I know a lot of computing now. And I guess technically I'm a computer science professor at this point. But I'm never going to really teach coding. I'm always going to be teaching biology.
Well, that actually brings up another question that I thought would be interesting to explore a little bit, perhaps. But so, in this field of study, you even just spoke to it. You came in from the biology side of things, the science side, but now, I don't know how you'd say if you're 50, 50, 60, 70, whatever. There's a lot of computer science running through your veins now, right?
There is, yeah.
And so your students, I assume, but actually maybe you could just share about that. Like, your students are coming in, or is it like 50-50? Some are coming in as really heavy in biology and others are really heavy in computer science, or how does that, how do those students come in? And if it is a mix, I mean, I know from working with computer programmers, there's a distinct culture. I don't know about in biology. I would assume maybe
there's a distinct culture.
They might not be the same culture.
They are not the same culture.
And so what's that like to try to do that? That's a challenge, right?
Biologists tend to be morning people, and computer scientists tend not to be.
Yeah.
So when I was in the biology department, I would stroll in at like 10 in the morning, and everybody else had been there since like 8, and I was the like late one coming in because I always teach in the evening.
Yeah.
I only teach graduate courses.
Okay.
But now that I'm in the computer science department, if I stroll in at 10, everyone's just coming in.
Yeah.
I mean, not everyone, but a lot of people.
Tends to be.
It's a later group. And when I'm leaving at 9, when in computer science, there's still a ton of people there.
Right.
When I left at 9 in biology, I would be the only one. Everything's all locked up.
Right.
But yeah, there's a lot of... There are different kinds of nerds and different kinds of geeks.
Yeah.
And actually, one thing that I've done is I teach a leadership course. It's called Leading Reproducible Research. But a big part of that is communication. And cross-cultural communication.
Right.
And I don't mean just cross-cultural communication between, people from China or Japan or Korea or, England or whatever, but also between biologists and computer scientists who think about things very differently. They're aware of very different kinds of errors and problems. And like as a biologist, I had no idea about things like silent error, which is where you have an error in a computer program that doesn't cause it to fail. It just gives you a result, but it's the wrong result.
Yeah.
I didn't know how to fix those or look for those until I started teaching, actually, because I wasn't formally trained as a computer scientist.
Right.
So part of it is making sure that everybody from both sides is aware of all the things that can go wrong.
Yeah.
So they communicate about those things.
Well, the cool thing about that is, and maybe this is me kind of romanticizing the field a little bit, but both sides kind of need the other side if they're studying with you, right? Like that's inherent in it. You can't just be dismissive of this group with their culture.
Right.
You need to understand if you want to be successful from your perspective doing what you're doing. So there should be invested interest in that, right?
Well, and I would argue that anyone who wants to be a successful bioinformatician really should understand both cultures.
Sure.
For comfort, if nothing else,
Yeah.
because you're going to end up hanging out with these
over
folks.
and over. Yeah. For many hours, right?
And so you need to
you're
learn
going to
the
want to
various
know
quirks.
that those guys are a little grumpy because they're not used to being up this early.
Exactly.
Or it's getting late in the day. I'm not going to get much out of those guys at this point.
Exactly.
You know?
There's definitely that. And so what we did, we developed it as a dual entry program where if you're coming in from a computer science background, you start out with a foundation course in biology. And if you're coming in with a biology background, you start out with a foundation course in computer science.
Okay.
So it gets you at least everyone to the same minimal level after that first.
Yeah, so you can start to talk to each other
Exactly.
with the language that you might not be familiar with.
Exactly.
Right.
Yeah. And then with biostatistics, both groups are kind of learning a little bit.
Oh, yeah.
And so that helps. And then we have some more advanced classes, both in the biology and in the computer science, that then, lets both groups become more sophisticated.
Right.
Because you really do need to know quite a lot of molecular biology in order to really interpret these analyses.
Yeah.
And you do need to know quite a bit of programming in order to develop these things to begin with.
Yeah.
So we want our students to at least be aware of everything that's going on.
So where would you say, this is kind of like a big 10,000 foot kind of question, but looking at challenges in bioinformatics, I know some other fields of science, they kind of go through these, it seems like they go through these cycles of challenges that are engineering based and challenges that are more like science knowledge discovery based, right? So trying to understand something, and then once the understanding comes, it's like, okay, we know this, but we can't execute. We need engineering to help us based on what we just learned. And so these cycles can happen, right? And so does that happen in bioinformatics? And if so, where would you say that the field is right now, or where you're at? Like, how would you describe that cycle? Is it we know so much right now, but we need to engineer so we can take advantage of it, or maybe the opposite, or how would you describe that?
Both are really, so what I would say we have is bioinformatics and precision medicine, whereas the bioinformatics is trying to understand what RNAs are getting made, or what are these, what functional importance do these have?
Yep.
And precision medicine is saying, well, if I have this mutation in, this gene, then this is going to happen clinically. I have a 98% chance of getting this kind of cancer or whatever. Right. And taking these things that we've learned and implementing them in a clinical standpoint where patients can actually, benefit. Thank you. I was trying to think of the word. Yep. And so we very much have an interplay between those two things, where there's biological discovery that happens, and then people try to come up with clinical tests that you can do to kind of make use of that discovery.
Yes. Okay.
So I would say both ways are kind of learning.
Yeah.
And then on top of that, we've been adding a lot more machine learning and sort of AI to the mix to try and find patterns that maybe we don't even understand why they're patterns, but they're reliable patterns that we see in the data, and what can we learn from those?
Yes.
And so that's interesting too.
Yeah. So that's kind of a mix, right? Because some of that is engineering, but then some of it is just pure science of like understanding data, right? Huh.
So for instance, a simple little tool. Well, it's not that simple, I guess, but they developed an AI tool that can look at pictures of moles and determine more accurately than most dermatologists whether that mole is precancerous or not.
Yeah.
Just based on the shape and so on. And I think they were even going to make an app for it where you can like take a picture of your mole and find out whether you need to go to the doctor or not. And it's actually quite reliable. So there's certain things they can do that with.
But they don't.
And then
with that one, do they know? Like, because a lot of times when I read about those things, like they don't even know how the AI is like it's definitely accurate, but they don't know why. Like they don't know how it's even doing what it's doing.
True. Yes.
Is that the case with this one as well?
I believe that's the case with this one.
Just wild, to think about. So,
Yeah, I mean, we know it has to do with the density of certain pixels.
Right.
We know we only gave it pictures.
Yeah.
But we don't know exactly how it can tell.
It's pretty amazing.
So there's a lot of that sort of thing. But then there's also a lot of big bioinformatics studies that are identifying a novel gene or, a mutation that's associated with a particular characteristic or illness or whatever. And then you come in clinically, and usually they just test for those few genes they know about. They don't do full-on sequencing. Or even if they do full-on sequencing, they don't tell you all the results. I recently I'm a cancer survivor, and so I recently had, all my cancer genes sequenced again. I had it done 10 years ago, but they only looked at a few genes, and none of those had mutations. And then when I had a recurrence recently, I had them look again, and they had a lot more genes that they were looking at. Yeah. But I couldn't get them to give me my whole sequence.
Oh, really? You're like, hey, listen, I can actually understand these things as opposed to the average patient, probably, right?
Right. Yeah.
So how has being a cancer survivor affected your study and your work and?
Profoundly, actually.
I would think so, but why don't you talk about that a
so
little bit?
I was actually a straight-up molecular biologist with aspirations of bioinformatics at some point. I was interested in bioinformatics because I did some bioinformatics studies during my Ph.D. years, but the actual analysis of it, we collaborated with bioinformaticians for. And Sarah Whelan, actually, Dr. Sarah Whelan, who's fantastic. And I talked to her about, how do I get into this field? I was at the very end of my PhD. I was, defending my thesis. And so she put me in touch with this group that studies schizophrenia and bipolar disorder. And that's how I went from being a yeast biologist who studies the regulation of transcripts, so RNA starts and stops in yeast, and instead studied schizophrenia and bipolar disorder, which, you can't really study in yeast. Yeah. So I switched over. And as I was learning neuroscience and much more complex genome and regulatory networks and so forth, I was developing some techniques, some molecular biology techniques for use in the lab in my first couple of months. And so basically I got this job as a molecular biologist, and I was supposed to stay a molecular biologist, but they had a bunch of RNA-seq data that they wanted to analyze, so they promised that I could learn some eventually. I could learn how to do these analyses. But in my first month of my postdoc, I got diagnosed with breast cancer for the first time. And I couldn't physically be at the bench after a while. So I did actually successfully develop the technique they wanted me to develop before undergoing surgery. So yay me.
For sure.
But it was still, a very rough way to kind of start this whole new field. And
it's a challenge in and of itself without having to deal with trying to fight cancer.
It was. I was already kind of drowning a little,
Yeah.
learning neuroscience and, all of this stuff.
Oh, yeah. Sounds like it might be a little bit difficult.
And I had a 3-year-old daughter at the same time.
Oh, my goodness gracious.
I was, overwhelmed. Wow. And then my poor PI, my boss, I was his first postdoc. We were just in the first month of my postdoc. He wasn't used to being a boss.
Right.
And unfortunately, at Hopkins, I wasn't a student. I counted as a student because I was a postdoc. So I wasn't an actual employee. So I didn't qualify for things like FMLA.
Oh, okay.
So there wasn't actually a mechanism for me to take time off for illness, or at least more than 6 weeks. So when I told my boss that I had cancer, his immediate response was, well, if you can come back eventually, I'll try and rehire you.
Wow.
And I said, I didn't say I was quitting.
Yeah.
I said, I have cancer. And he said, well, you still need to be productive. He was very nice, actually.
Right.
He was very supportive. My whole lab really was, for the level of lab that it was and the level of researcher I was and so forth.
Was that just kind of for him, like it was a reality of the lab?
It's a reality in every lab, honestly, that you have to publish.
Yeah. That's pretty tough.
It's very tough.
Yeah.
And it was a reality for him that he had to publish.
Right.
And, and he couldn't publish unless I published. And,
Okay.
He could publish without me, but, my publishing definitely helped him and so forth. But anyway, they ended up keeping me on, and my way of being productive was then to Sarvan Sabanti, my PI, got me involved with Jeff Leake, who's this great bioinformatician, and he, out of the kindness of his heart, said he would help me learn bioinformatics. And so Sarvan and Jeff gave me some data and pointed me in the direction of some tools. And I just learned by Googling. And so I taught myself Unix, Linux, and I taught myself R.
Wow.
And I taught myself some biostatistics. And as I was doing it, I was working on real data and just kind of getting to a certain place and getting stuck, and then Googling and getting somewhere else and getting stuck, and then Googling and so forth. And there's a lot of great online communities like BioStars, and Stack Overflow is for all programmers, basically. But I spent a lot of time on Stack Overflow and a lot of time on BioStars. And then when I came up with my first script that did my full analysis, I brought it to Jeff Leake. And I still remember sitting there, and he's reading over it and reading through it. And he looks up and he says, "This is surprisingly reasonable for a biologist."
And that was for him. That was a genuine compliment.
For me, it was a genuine compliment. I was thrilled. I was reasonable. Yes. And of course, over time, I've gotten a lot better.
Right.
But I could come up with a kludgy solution. It might be 300 or 400 lines of code when it only really needed to be 25. But I got there in the end, and it was right.
That's from my experience, actually, I was just talking with one of our developers today. That's part of it. Anytime you write something, you look back and you go, I could do that better. And you do it better. And then you look at it again, I could do that better. That's the nature of learning to code and getting better. So,
Right. Yeah. And I think that's part of how, in the larger sense, these tools are improving over time. I think they're going to keep converging on each other. Over time, we're going to find the answers because everyone's iteratively doing better and better.
Yeah, once you have it there and it's concrete, you can look at it, you can kind of, pick it up, turn it around, look at it from different angles and go, oh, I can make this better now that I see exactly what it is. Right?
Exactly.
Yeah. So how does, so that's how cancer had a huge impact on you getting into the field.
Right.
And how does it impact you now as far as thinking about what you're doing and your approach to it?
That's a good question. So in the to finish the first part,
yes.
I never really went back to the lab. I mean,
Okay.
I did to train students, but I never went back to, I mean, I did some biological studies in the lab, but my main focus after that became analysis, because by the time I was back on my feet and good, I was actually good enough to do these analyses. Wow. And we had all of this data. But the way it affects me now, especially with my recurrence, has been difficult, because I went through a lot of chemo last year. Yeah. I actually didn't stop teaching completely, which was maybe a mistake. I had a few capstone students that I wanted to get them through their projects, so I kept teaching them, because I wanted to make sure no student would not, that it wouldn't be the case that a student wouldn't graduate on time because I got sick. Wow. And so
that's dedication to your students.
I am very dedicated to my students. I love my students. I love teaching. And I guess maybe the way my illness has affected me more than anything in recent years hasn't been so much about the research that I do, except maybe that it's impeded it a bit. Yeah. But the way I deal with students and kind of focusing what's important to me. And you know, what's important to me is teaching, you know, having a legacy of students who are self-actualizing and, you know, embracing their futures. And one of the things that I love about bioinformatics is it has in many ways democratized science, in that there are, yes, you need to have access to a strong computational tool, but there are a number out there that you can use for free. So you can go online and do an RNA-seq analysis. It might take you a long time, but you can use publicly available tools to do the whole thing.
Why is that? I mean, I can think of why I would think it would be exciting, but why is that exciting to you? Like, why do you think that's important?
I think it's important because that allows people who aren't at Johns Hopkins or, you know, University of Washington or, you know, someplace like that. Right. It allows them to ask just about any biological question. I mean, lots of biological questions. I mean, I have trouble, you know, narrowing down the questions I want to ask. And you can get answers, you know, real answers. And you don't have to be at a major research institution in order to do that. It allows me at Hood, with my resources, which are not great, to be honest. I mean, we're a teaching college. We're not a research institution. And you know, it's often assumed that you're going to have $100,000 in research funds to do these kinds of projects, and we get $400 a year. Wow. You know, you can't do a lot with that. And so, but I can, because I can access these publicly available databases, and so I can just use my imagination and these tools and the Hood cluster, and answer these questions.
Well, I would think, too, there would be, I might be wrong on this, but there might be a pressure at those institutions to study certain things and to look for certain answers and study certain problems, because they're the, quote, big problems. And that's good. Like, you want that. But if you don't have that pressure, you can ask different kinds of questions that maybe don't seem consequential, but turns out, oh, this one was consequential, but nobody was asking this question. Like, that can happen because of that democratization, right?
Exactly.
Which is really exciting.
Exactly.
So, yeah.
Yeah. I had a student with a particular disorder, in fact, that's relatively rare and not much is known about it. And we came up with some really interesting questions surrounding her disorder. We ended up not having enough publicly available data to answer the questions. We tried, but I'm in a few years, I want to ask those same questions again. I think we'll have enough data.
And just the idea of that is really exciting, right?
Yeah, it is.
Somebody can ask that question that wants to ask it, you know, and it doesn't have to be in that institution that maybe is a little less accessible. So wow, that's really cool. So how did you know, COVID seems to have affected everybody in different ways, and we talked about this a little bit beforehand. I was It was really interesting, your answer. So share with us, how COVID affected you all in the program.
It was a challenge, as it was for everybody, especially since my students are all graduate students. So many of them have kids at home. Most of them are working full time. And so even just pivoting to online instruction was still very difficult for them because they have kids at home, interrupting them and so forth, and just kind of not enough bandwidth. Yeah. So we did lose some students they were just too overwhelmed with full-time care of their children plus full-time work plus going to graduate school. But another thing that kind of hurt us, I think, so we did read, our program was really doing very well so far as our numbers, and we were getting ready to start having, summer versions of classes and, offering extra versions of classes and offering extra, new electives and things. Because we had enough students to really make that happen. But it's fallen a little bit because one of the things that I love about Hood is we're face-to-face. And there's a lot of online instruction and ways to learn bioinformatics online that are better or worse depending on, where you go. And might be more expensive or less expensive. And the students that we tended to get are the students who really value having a face-to-face interaction, who value the fact that we have small class sizes because we have about 8 to 15 students per class.
That's great.
And lately it's been between 5 and 8 per class because we dropped down a bit. And so they like the fact that they get to know the professors get to know them. We have a mix of Hood faculty and also adjuncts, all of whom work at the National Cancer Institute or at biodefense labs or something like that. Right. So it's a good way for our students to get internships and jobs.
Because they're going to probably make some close personal connections there, and that's going to be really helpful.
If you're in a class of 5 students, it's very easy for the professor to get to know you.
Sure. And
so that is a big value that we add. And so I think that's part of why we saw a decrease during the pandemic. But also what happened at the same time is everybody who was a bench scientist couldn't be in at their bench, at least for a while. And so everyone wanted to analyze their data all at once. And so the other thing that happened was I had some more advanced students who were getting close to graduating, but they just left to go be bioinformaticians because the field is so hot that they didn't even need to finish their degrees. They just had to prove that they knew how to do.
If they could do the work.
Yeah.
Get out there and do it.
I can tell you my students who do finish their degrees are getting placed very well.
I'll bet.
And very quickly. They all make more money than I do doing this. You can make a lot more money doing bioinformatics than you can teaching bioinformatics.
Wow.
But as I said, my reassessing my priorities, I actually really love teaching. I get a lot of satisfaction out of it.
Well, I mean, I'm sure you've thought about this, but as you're describing that, it's amazing the impact that you're having, right?
I hope so.
I would think so. Sending these students out, I mean, they're learning from the program, that you're building or built, and who knows where they're going to go, what they're going to do, but there's a multiplying effect there, right?
Oh, absolutely.
That's exciting.
And I've been able to send, some of my better students. Well, actually, I mean, I shouldn't say better or worse. They're all good in their own, unique ways. Yep. But I've been able to send a number of students to graduate school, Ph.D. schools, or to do. Several people have gone to Hopkins to do research, get positions. Several people have gone to NIH to get positions.
That's cool.
So that's gone well.
Yeah.
And the other thing I really love about Hood And this kind of fits into what I was saying about the democratization of science, is Hood is a very democratic place. Even though we're just outside Fort Detrick and the big research labs there, we're very much a teaching-focused place. And so we actually accept people with sort of lower GPAs because we feel confident that we can, teach people, especially students who come. I accepted a lot of students who come in who academically weren't great when they were in undergrad, but have gone on to the workplace and learned a whole lot and matured a whole lot. And they come in and they just flourish with me. And, they may not be able to get into an Ivy League school, but I can get them to the point where they can compete with the people coming out of those schools.
That's awesome.
I mean, I believe I can.
Yeah.
They are competing successfully.
So you have done it.
I have done it.
Yeah.
And so I love that. I love that. When I was at Hopkins, I tutored. I tutored and I also trained some undergraduate students as I was going through. But they tended to be students who their parents were already doctors. They already know how academia works. They already had straight A's. The reason they got the chance to come and work with me was because they already had straight A's, they were already ahead of the curve, et cetera. And those students, they were wonderful, but they're going to do well no matter what. And what I really love about teaching at Hood is I can take people, as long as they're interested and willing to do the work, I can take students who aren't on that trajectory already, and I can get them on that trajectory if they want to be. But
it's got to be very satisfying.
It is, yeah. It's really what I most enjoy about what I do.
That's great. So, again, this is kind of a big picture thing, but if you look at the advances in the field and you think about kind of where things are going, might be maybe really near future, like in the next year, or might be further out, but what are things that you're really excited about in bioinformatics that you think are going to be, and again, if you can, try to, to the average person, the ripple effects that we're going to see, right? What are the things that you're thinking, oh, man, these are the things that are coming that are just amazing?
So one of the things that I think is really exciting is shotgun proteomics is becoming, and metabolomics. There's a big word there. Yeah. So proteomics is just the study of proteins.
Okay. All right.
And metabolomics is the study of metabolites.
Okay.
But using mass spectrometry, you can basically take a complex sample of proteins, let's say a chunk of, or several blood cells that someone has drawn and digested down to their proteins. You can run that on a mass spectrometer and try and figure out all of the different proteins that are in those cells. Okay. Now, there's some successes with that and some failures with that, but I think that's going to be a new thing for bioinformatics. I think they're going to come up with better ways of So they have good ways of breaking down the proteins into little tiny pieces and finding little patterns of peaks and valleys that the machine makes that go along with those particular proteins. But that's just a little piece of a protein, and these peaks and valleys, might come from a different protein than these peaks and valleys. And trying to put that all back together, just like I said, putting 100 base pair sequences back together, putting proteins back together is an even harder part, so understanding all of the different pieces of protein that are in there is challenging, but I think it's coming.
So if they could do that, what does that translate to?
So for me, the way that translates is if I find a novel RNA that's not annotated anywhere as being an RNA that gets made in humans, but I see peaks in the RNA sequencing that make me think, okay, this RNA does get made even though it's not supposed to be. Then I can go and I can look for the protein and see for sure, is it made or is it not made? Because DNA gets translated into protein, RNA gets translated into So DNA gets transcribed to RNA, RNA gets translated into protein, but not all RNAs get translated, and you don't know to the level to which. So that would let you know really what's going on. It's a different way to get the picture of what's going on in the cell because it has different biases, different things that go wrong.
Okay. So it could just paint a more full picture.
If you have both, it paints a whole picture.
It's like a higher definition of what you're looking at, maybe.
Exactly.
Okay.
The other thing that I'm excited about is I think we're going to increasingly be making tools where bioinformaticians are making tools that output data that can go into simpler tools. I'm trying to generalize a little bit, but I think bioinformaticians are going to be able to come up with tools that we're using and conclusions that we make doing bioinformatics that then can be translated over into precision medicine. And we can basically make the tools easy enough to use that a clinician can use it. So, we'll do our sequencing analysis pipeline and refine it to the level where, if you want to screen for certain kinds of conditions, you just run this pipeline and it'll spit out a yes or no answer or a probability answer. I mean, I think that's going to happen before too long.
That's exciting. Huh.
And so more and more clinicians, not just the people at the big universities, but more and more clinicians are going to be able to have access to the ability.
Tools to diagnose very specific things.
Yeah.
Okay.
And at the same time, the sequencing technology has also been improved quite a lot. Okay. In fact, one of the new things is they've really improved long sequencing now. So now instead of 100 base pairs at a time, now I can get 800 or 1,000 base pairs at a time. And that, then it used to be we could do that, but it was really inaccurate, so it wasn't worth doing. Yeah. But now it's getting more and more accurate. Okay. Not because of the sequencing itself, but because of the analysis tools that they've come up with. They're starting to come up with better heuristics and stuff to figure out if these things don't agree, which one's right, sort of stuff. Cool. And so I think that's going to lead to a big boom in understanding which RNAs get made out there. And so, all of that together is going to teach us a lot about how bodies work and, how to find and interpret things that are maybe not working.
Yeah. Do you foresee that becoming, so a lot of what we've talked about, what you've described is, these data sets that are kind of, from how you've described it, kind of far removed from me, the person, right?
Right.
Do you think these tools and the study, the analysis will get closer to a person, real time, like your genome?
I think that is going to happen. Yeah. I want to say 5 years, but I might be wrong. Right. Part of it, we're already able to do that. Now it's just a matter of, the big thing is interpretation. Okay. So part of the reason they wouldn't give me my own DNA sequence is they don't want me to find stuff that might be disturbing to me. Okay. I pointed out I've already had cancer twice. I don't think that learning that I have a predisposition to cancer is really going to upset me at this point.
Yeah, exactly. Could have guessed that one.
But they don't want to give people information they don't know how to process.
Sure.
And they don't want to give people information that might be disturbing to them that they can't do anything about. Yeah. So if there's nothing we can do to cure you, does it really help you to know you have this mutation? Right. Or does, especially...
And really, the answer to that depends on the person, right?
Right. Well, and it depends, it depends on what the mutation is, too, I guess. I already have cancer, so the reason I wanted to know about my genes is for my daughter, right? Yeah, sure. So for those purposes, it's good to know. But basically, they need to figure out a way to add some sort of interpretation and counseling component to these tests. And, that might be a whole new range of nurse or
Right.
counseling,
Yep.
Not necessarily nurse, but clinician of some
Yes.
kind who's, they already have genetic consultants.
Yes,
But that
I was
might
say they're
be
starting
something.
to, might be a growing field, I guess.
I believe
right?
it will be,
Yeah.
actually.
Right. So that person would kind of sit down with you, go over your results, and understand how to explain it in a way that helps you make better decisions.
Exactly.
Or not make a decision, depending.
Right.
Yeah.
And I think there should be people like that on staff at physicians' offices or at least hospitals for the physicians to chat with as well.
Right.
People who really
right.
understand,
Yeah.
if you have this mutation, what does that really mean?
Yeah.
Because it can be very challenging. Like, even if you have a breast cancer gene, like BRCA, BRCA1 and 2 are well-known breast cancer genes, and there's many, many different mutations, so they know exactly what that's going to do to you. But there's lots of things, you have a mutation in that gene, but we don't know what that would do functionally.
Huh.
Like, it's not clear
Yes.
how that's going to affect anything.
Right.
So
So it becomes a challenge.
it becomes quite a challenge.
And even the things that we know are changing all the time.
Right.
And so for a doctor, like, who's meeting with people daily, for them to keep up with that,
They wouldn't
already
have
have
time.
a lot to keep up
Right.
with, right? This is a field that's changing super fast.
Right.
So to figure out that interaction with an expert at key moments, something is going to be important for that to develop further, right?
Mm-hmm. Absolutely.
Yeah. Huh. Okay. Yeah, I'm actually hoping to, in future shows, be able to bring in some people to talk about precision medicine. 'Cause that to me is, kind of the other side of a lot of the things
Yeah,
you've been talking
it
about.
is,
right?
absolutely.
So it's a really interesting field.
And it's worth noting, I don't know if it'll get passed, but I've been on a committee that has designed a health informatics program
Okay.
that we're considering offering at Hood. We just presented it to the graduate
interesting.
program. So it would be a master's in health informatics.
Okay.
And it would share a lot of the same computer programming stuff, machine learning and Python and R and
Yep.
data science, essentially, that my bioinformatics students will get. But then instead of the biology, you'd be getting clinical study type stuff.
Okay. Oh, that's pretty interesting.
So, yeah, there's a lot of this can morph a lot of different ways.
Yeah.
But I think the bottom line is that biology is infinitely complex and, to my mind, infinitely interesting.
Yeah.
And no matter what we learn, we're just going to keep having more questions and more questions and more questions and more questions.
Yeah.
And I love that, actually.
Yes, I can tell. That's one of the things for you that makes it so exciting.
It really is,
Yeah.
that there's still so much to discover.
Yeah, that really is kind of the essence of science, right?
It
that
is.
excitement of discovery.
Yeah.
So, and you're in the thick of it, for
I
sure.
am, yeah.
I was going to say that it's got to be exciting to be involved in this field now.
It really is.
It seems like a pretty unique time.
It's a very hot field and it's growing, and that makes so much opportunity. So I had another one of these students who he's awesome. His name's Connor Jenkins. He's doing a Ph.D. program now. But he kind of came in not feeling so good about his academics, but had some work experience and kind of came in. He wasn't sure if he would even get into the program and stuff. And we talked, and I talked to him, and I immediately knew that he was smart and creative and, a problem solver. And I didn't really have any qualms with accepting him into the program.
Right.
And he did flourish. But in his first, he was a mass spec technician, kind of expert. He set up and operated the mass spectrometry instruments.
Okay.
And in his first year in the program, actually after his first semester, he wrote a new algorithm for analyzing mass spectrometry data. He came up with a new way, way to look at the data, essentially, and published it in that first year.
Yeah.
So, even someone who thinks he's coming in as a goofball,
Right.
did really well.
Yeah.
Because there's there's so much stuff we haven't figured out how to do yet.
Right. And that goes back to that democratization, right?
Exactly.
It's accessible to this guy,
right,
where it might not have been before.
Exactly.
And he's the guy that needed to be able to do it. Right?
Exactly. Yeah.
Right place, right time.
Yeah.
Yeah.
Exactly.
Wow. It just makes you think what the future, the near future holds is going to be very exciting.
It really is.
We might have to do another interview in a year or so.
Oh, well, I would love that.
Yeah.
Yeah, that'd be great.
Yeah. Okay. Well, I always like to ask, and I don't know, anything that something you like about Frederick, as far as anything you like to do downtown, or things you like to do in your.
I love
Frederick.
If
and when you have free time.
Well, actually, moving here was a huge game changer for me.
And where were you?
We were in Baltimore. I was at Hopkins for 14 years.
Oh, you were for quite a while.
Yeah,
okay.
I started as a technician, and then I got my Ph.D. there, and
okay.
then I did my postdoc there, all in different departments, all on different topics completely. It could have been 3 different institutions, but wasn't.
Yep.
And so we were in Baltimore for quite a while.
Okay.
And we moved to Frederick, and because of my medications I was taking before for the cancer, I was still on some medications that gave me terrible migraines, and I couldn't drive.
Oh, okay.
And we lived 45 minutes from where I worked, and I was carpooling with my husband, which was fine, but then his job got moved to Columbia.
Oh,
and Johns Hopkins, the medical campus, is in eastern Baltimore, kind of a dangerous area of Baltimore. Anyway, so once I came to Hood, it was fantastic because I don't have to drive. So I couldn't drive because of the migraines I get. I see circles around
ocular
lights.
migraines.
I see halos around every light.
Yep.
So if I'm driving at night and it rains, I see a halo around every drop on the windshield.
Oh, wow.
So I just can't see.
Yes.
So that's why I don't drive.
Yeah.
But being Hood and living downtown, I can walk.
Yeah.
I could walk my daughter to school every day. She went to Parkway Elementary, which was a fantastic elementary school.
Yeah.
And then I could walk from Parkway to Hood. And
beautiful walks.
Beautiful walks.
Yeah.
And then I could walk back to Parkway and pick up my daughter and walk her back to Hood. And for the first several years I was in the biology department. The biology department has tons of mothers and grandparents.
Huh.
And so they were totally okay with me bringing my daughter.
Oh, wow.
They'd bring
That's
her in, show her a dissection, or bring her into the greenhouse and have her help in the greenhouse or whatever.
Wow.
So
like
one of the things
atmosphere
I've
there.
loved, has been Hood. I mean, Hopkins is great in its own way, but Hood is so welcoming and loving and caring.
Yeah.
I found it to be just such a welcoming environment. So I love that. And I love being downtown and getting to eat downtown and, walk around downtown and happy hours and stuff. It sucked having COVID because of that kind of, because I'm still doing chemotherapy, I don't go I don't do indoor dining or anything. So I've missed that.
Sure. Now, are you introverted naturally or extroverted?
I'm actually naturally introverted.
Okay.
I've had to force myself.
Okay, I wouldn't have guessed that.
Well,
so you're doing a good job of you definitely.
It's a complete act.
Yeah.
I've really
your
had to force
natural
myself.
inclination is in data, doing things.
Oh yeah,
yes, okay, right.
Navel gazing, and
Yes.
the data person. In fact, in my family, I'm known as the one with no social skills. Which is really funny now that I'm in the computer science department, where I'm the one with social skills.
Let's say you got to send your family this video and say, "See, I got some social skills."
I'm kidding. There's actually many people with good social skills in our computer science department, but we joke about it because
of
computer
course.
scientists generally aren't
Yep.
the talkative ones.
Yeah. Well, I just want to say, thank you so much.
Oh, thank you. Thanks for your interest.
Yeah. I mean, it's been a great conversation. It's really interesting to hear what you're doing at Hood, and just your story of how you got there and your passion for teaching. I'm excited to know that's happening there and that you're doing what you're doing. So thanks for sharing that. I'm sure a lot of other people would be really interested to hear this as well.
Well, thank you. And I'm excited you're doing what you're doing. It's great to, bring light to all of the great stuff that's happening. I was watching your blog, and there's a lot of stuff I didn't know was going on here in Frederick. And I thought I was in touch with the tech community. So, clearly there's a lot going on I didn't know about.
Yeah, that's one of the fun things about doing this is it does, open doors for me to have conversations with people I wouldn't have been able to otherwise, which is what it's all about. Lots of fun, for sure. So, all right. Well, thank you.
Thank you very much.
All right. I hope you enjoyed the show, and I hope it gave you a new insight into the talents, passions, and curiosities from Frederick, Maryland, and beyond. If you did, maybe you can help me out a bit. The best way for the show to grow in its reach is for you, the audience, to subscribe, like, and share this episode with your friends. Much appreciated. Until next time.